Our study was focused on vascular amyloidosis and how it impairs the gliovascular unit in a mouse model of Alzheimer's disease.
This is a cartoon depicting the gliovascular unit with astrocyte end feet expressing the water channel and vascular smooth muscle cells, or pericytes, surrounding the endothelial wall. This unit is composed of neural and vascular cells, assures sufficient blood supply to active brain regions. Astrocytes, vascular smooth muscle cells, and pericytes are important players in modulating vessel diameters. The reduction of cerebral blood flow in patients with Alzheimer's disease may thus cause ischemic damage to neurons, suggesting that neurovascular dysfunction may contribute to Alzheimer's disease initiation and progression. In the normal brain, glial, neural, and vascular cells cooperate as a functional system called the gliovascular unit to ensure continuous blood supply that correlates with neuronal activity. Here, we studied how the gliovascular unit is affected in a mouse model of Alzheimer's disease using a combination of ex vivo and in vivo imaging approaches.
We labeled vascular amyloid in living mice using the dimethoxy-X04. We elicited vessel responses in ex vivo using pharmacological stimuli in vascular smooth muscle cells, or astrocytes. Multiphoton in vivo imaging through a cranial window allowed us to complement our ex vivo data in the presence of blood flow after label-free optical activation of vascular smooth muscle cells in the intact brain.
We found that vascular amyloid deposits separated astrocyte end feet from the endothelial vessel wall. High-resolution 3D images demonstrated that vascular amyloid developed in ring-like structures around the vessel circumference, essentially forming a rigid cast. Where vascular amyloid was present, stimulation of astrocytes or vascular smooth muscle cells via ex vivo calcium uncaging or in vivo optical activation produced only poor vascular responses. In addition to its toxicity to neurons, amyloid B may also influence the cerebral vasculature. Amyloid B may accumulate in the walls of cerebral vessels, a pathology termed cerebral amyloid angiopathy or vascular amyloid, and these deposits can narrow the lumen of vessels and induce vasoconstriction. In addition to its toxicity to neurons, amyloid B may also influence the cerebral vasculature. Amyloid B accumulates in the walls of cerebral vessels, a pathology termed cerebral amyloid angiopathy or vascular amyloid, and these deposits can narrow the lumen of vessels and induce vasoconstriction.
Already high during rest, oxygen and glucose demands increase even further in active brain regions. These constantly occurring spatial fluctuations in energy demand require a similarly flexible adjustment of the cerebral blood flow. This process is called functional hyperemia, or neurovascular coupling, and is regulated by the GVU. Astrocytes glial cells in the brain and spinal cord are ideally positioned within the GVU to mediate functional hyperemia. They have two types of processes: large perivascular end feet that cover the entire cerebral vasculature, and finer processes enwrapping neuronal synapses. Astrocytes respond to neuronal activity by increases in intracellular calcium. This causes the release of vasoactive factors that act on the contractile cells within the GVU, vascular smooth muscle cells, or pericytes, to modulate blood vessel diameter. Using a combination of in vivo and ex vivo imaging of the GVU and the HAP-J20 male and female mouse model of Alzheimer's disease, we found abundant amyloid deposits on blood vessels, age-dependent cortical reactive gliosis, and loss of astrocyte polarity. We used electron microscopy and immunohistochemistry to view and analyze sections of the mouse brains.
We used statistical tests to analyze our results, and all data reported in the figure legends or results. Outliers were excluded from the analysis were noted.
In patients with Alzheimer's disease, amyloid B accumulates around the cerebral vasculature. This vascular amyloid deposition is replicated in mouse models of the disease. To better understand the physical relationship between vascular amyloid and its associated vessels, we created high-resolution 3D reconstructions and volumetric optical section projections from in vivo and ex vivo multiphoton and confocal data. Three-dimensional reconstructions illustrate how the bands of amyloid can span the entire circumference of vessels in some areas but not others. As the disease progresses, the gaps shrink and the vascular amyloid starts to form contiguous structures.
We found that amyloid deposits mostly displaced astrocytes, and the end feet no longer abutted the arterial elastin layer in areas where vascular amyloid was present, as seen in Figure 2b. However, there were a few locations where the end foot was still in its normal vessel juxtaposition, as seen in non-transgenic animals. Using video microscopy analysis, we measured changes in arterial diameter in response to bath application of norepinephrine in acute slices from control and HAP-J20 mice. As expected, most arterioles in control slices dilated in response to norepinephrine, and a paired analysis indicated a highly significant effect of TACPD. However, in HAP-J20 vessels, there was no significant effect of TACPD in a paired analysis, suggesting impaired dilation of vessels in HAP-J20 mice in response to TACPD.
To visualize vascular amyloid, we injected methoxy XO4 12 hours prior to sacrificing control or HAP-J20 animals and used confocal imaging to identify vessels with or without vascular amyloid. In acute slices from control mice, calcium uncaging in astrocytes led to an immediate rise in calcium followed by biphasic vessel constriction with an immediate constriction followed by a slower sustained vessel response. In slices from HAP-J20 mice, encaging in astrocytes near arterioles resulted in similar increases in calcium. To determine if the impaired vasoconstriction in HAP-J20 mice was due to the amyloid burden on the vasculature, we compared the vasoconstriction of arterioles with and without vascular amyloid. In HAP-J20 mice, the response of arterioles without amyloid was comparable to arterioles from control mice. In contrast, the presence of vascular amyloid significantly decreased vasoconstriction in both the initial and the maximal response.
Astrocytes release vasoactive substances that stimulate contractile VSMCs or parasites on arterioles or capillaries, which then constrict or dilate. To assess these parasite functions directly, we use drugs that bypass the astrocyte component of the GVU and instead stimulate VCSMCs directly. Using video microscopy, we measured changes in arteriole diameter in response to bath application of ET1. Arterioles from HAP-J20 mice had significantly less vasoconstriction in response to ET1 compared to control mice. Taken together, these data suggest that the ability of parasites that properly function in regulating vessel diameter is impaired in HAP-J20 mice.
Vascular amyloid dampens the vessel response in vivo. An important next step was to examine potential changes in VSMC function in vivo, where blood flow is present and vessels are pressurized. For these experiments, we adopted minimally invasive in vivo multiphoton imaging. We visualized the vasculature and amyloid through a chronic cranial window in live mice. VSMCs were identified by selecting arterioles positive for alexaflor 633 hydrazide and placing the stimulation focal point at 4 mm distance from the border of the vessel lumen, as previously described. We compared the vasoconstriction of arterioles with and without vascular amyloid in HAP-J20 mice. In vivo, we observed a rapid response immediately after stimulation of the VSMC. The observed change in vessel diameter was comparable to the maximum response observed after pharmacological stimulation of VSMCs in brain slices. Areas with vascular amyloid constricted to a lesser extent than areas just downstream of the amyloid on the same vessel. The importance of astrocytes in regulating local blood flow to match neuronal activity is now well recognized, and the functioning of the GVU and the associated signaling cascades are well documented. However, surprisingly little is known about the GVU in disease. This is particularly surprising since most neurological diseases present with relative reactive astrogliosis, which might profoundly alter the relationship between astrocytic end, feet, and blood vessels. Consistent with previous reports, we saw the progressive development of astrogliosis characterized by increased JFAB expression and changes in cell polarity defined by redistribution of AQ4, which is enriched in astrocytic end feet. Using multiphoton in vivo imaging with the amyloid blinding Congo red derivative methoxy XO4, we showed a direct association of amyloid with the endothelial vessel wall and confirmed by electron microscopy that amyloid is deposited between the astrocytic end feet and the blood vessel, essentially separating the end foot from the vessel wall. These data could be interpreted by amyloid altering the VSMCs, or alternatively that the amyloid deposits can act as a rigid physical exoskeleton that prevents the vessel from changing its diameter. Segments devoid of amyloid showed normal responses, yet a few hundred micrometers along the same vessel in an area with pronounced amyloidosis, constriction was significantly reduced. This is an important point when considering amyloid clearance strategies or immunotherapy. If amyloid deposits are principally responsible for the impairment of the vasculature, an important question pertains to the reversibility of the amyloid-imposed impairment. Can dissolution of vascular amyloid restore normal vessel function? Our study suggests that there is a time window for therapy during which amyloid clearance would allow the vessel to regain its full dynamic range.
Our findings illustrate clearly the physical hindrance that vascular amyloid poses to normal regulation of vascular tone.
One of our critiques would be that the mice were all of very old age, and the more imaging and staining techniques would have been helpful to better imagine what it actually looks like.